The topic for this lab presentation is meiosis, a process of cell division that occurs in the reproductive system. Your learning objectives are listed in the gray packet of lab handouts on page 53. Additional resources to help you understand meiosis are available in section 17 of the green lab manual and in your textbook on the pages listed on this slide. Sexual reproduction involves two processes, gametogenesis and fertilization. Gametogenesis is the process by which gametes are produced. In females, the gametes are called ova or oocytes, commonly referred to as eggs. In males, the gametes are called spermatozoa, commonly referred to as sperm. Fertilization occurs when a sperm and an ovum join. The resulting single cell is called a zygote. A multicellular organism then develops from this single cell throughout growth and development. Gametogenesis in females occurs in the ovary and is also called oogenesis. Gametogenesis in males occurs in the testes and is also called spermatogenesis. Although there are some differences between oogenesis and spermatogenesis, both occur in the gonads and both involve the cell division process called meiosis. During fertilization, the genetic material present in an ovum unites with the genetic material present in the sperm. Therefore, each will contribute half the number of chromosomes to a zygote. In human somatic cells, there are 46 chromosomes, or 23 pairs, shown here in this human karyotype. 46 is called the diploid number because there are two copies of each chromosome. Germline cells that are present in the gonads are also somatic cells, and they will be the ones that divide by meiosis to produce gametes. Therefore, germline cells are diploid cells and they have 46 chromosomes. Gametes are haploid cells and they have 23 chromosomes. A gamete has only one copy of each chromosome pair. This is called the haploid number and of course equals 23 in humans. Recall that mitosis produces two daughter cells because each chromosome in the parent cell, two diploid daughter cells, because each chromosome in the parent cell is replicated before the cell divides. Replication will result in a chromosome having two chromatids joined at a centromere. During the process of mitosis, which you studied in A&P 1, this centromere splits, and one chromatid will go to one cell, the other chromatid will go to a different cell. In meiosis, the process will be slightly different. Replication will still occur. A single chromosome will undergo replication, and two chromatids will be attached at a centromere. But in meiosis, we will have four products, four separate cells resulting, and each cell will have one single chromatid present. The diploid shown in this diagram has two pairs of chromosomes. So if this is a germline cell, it will undergo meiosis to produce four cellular products. This diploid cell has a diploid number of 4. You have two red chromosomes and two blue chromosomes. The products are haploid cells, and notice there are only two chromosomes present in each cell. There will be two divisions. Meiosis I will happen and then meiosis II. First, replication occurs, just like in mitosis, and now chromosomes are composed of two sister chromatids each. After the first meiotic division, you'll notice that the two longer chromosomes are no longer in the same cell. They have been separated. They are homologous to one another, and so this indicates that homologous chromosomes are separated from one another. In meiosis II, sister chromatids will separate. So one strand will split from the other strand as the centromere divides. The products of meiosis I are considered haploid because there are no longer homologous pairs. The products of meiosis II are also considered haploid, although each chromosome is no longer composed of two chromatids, only one chromatid. So in summary, the features of meiosis include that the process occurs in diploid germline cells. There will be two divisions. There will be 4 haploid cells produced, and, most importantly, each daughter cell is genetically different, not just to each other, but also genetically different than the parent cell. The process of meiosis involves a complex orchestrated movement of these chromosomes to have the distribution occur correctly into the haploid cell products. So we will trace the movement of chromosomes during the first division, that's called meiosis I, and during the second division, that's called meiosis II. The stages are similar, familiar to you as what you learned in mitosis. The stages consist of prophase, metaphase, anaphase, and telophase. It's just that they occur twice. There are a few differences that occur in each phase, particularly during meiosis I, the first nuclear division. In prophase I, a unique phenomenon occurs in which homologous chromosomes align. This is called synapsis. They pair up together. And here I have a picture of 2 replicated chromosomes that have lined up, the centromere shown in red. Homologous recombination occurs shortly after. This is also known as crossing over. And sister chromatids will actually physically move, break apart, and then go back together. So they exchange chromosomal material. What this does is change the genetic composition of each chromosome. It gives a lot of genetic diversity to the human race. In metaphase I, after synapsis and homologous recombination has already occurred, the chromosome pairs line up at the metaphase plate. In this cell, we have a diploid number of 4, or 2 pairs of homologous chromosomes. How this pair lines up with the red on the right or the blue on the right, either way, could be 180 degree flipped, will not dictate how this pair lines up. This is called independent assortment, and it's the basis for how genes are inherited. Down at the bottom of the screen, you'll see that there's a picture of the metaphase stage of mitosis. And notice that the chromosomes are all lined up singly, not as pairs. In anaphase I, homologous chromosomes separate. So one chromosome will go to one pole, the other chromosome will go to the opposite pole. In telophase I, cytokinesis occurs, and a nuclear membrane forms around the chromosomes. The 2 resulting cells are called haploid cells. Even though that might not make sense to you yet, it's because there's only one homologous chromosome remaining. Only one copy of each chromosome pair. However, That chromosome, the long one for example, consists of 2 chromatids. In meiosis II, what will happen is very similar to mitosis. We will start with a cell that we consider haploid, and it will divide through the stages of metaphase, anaphase, and telophase to produce 2 cells. But remember, there were 2 cell products from meiosis I. So in the end, we get 4 products total. And each chromosome has lost 1 chromatid, so to speak. So there's only a single chromatid present in each chromosome. The process of meiosis happens slightly differently in males than it does in females. So in males, spermatogenesis follows this typical pattern where we get 4 final products. Each are haploid, and they are all viable. So all 4 haploid sperm are viable. In females, the process is different. The division of the cytoplasm in both telophase stages is unequal. So cytoplasm is distributed to just one of the oocytes. And therefore, we call that one product an oocyte, and it is viable. It will live. There are 3 products that will just contain genetic information, having a nuclear membrane surrounding the chromosomes with very little cytoplasm. They are still haploid, but they are non-viable. Without mitochondria or any of the nutrients present in the cytoplasm, they are non-viable. In addition, meiosis II arrests. It will only continue and complete if fertilization occurs. This slide summarizes the difference between spermatogenesis and oogenesis. At the beginning of spermatogenesis, we have a germline cell. It is diploid. It undergoes metaphase I to produce 2 haploid cells. Those cells will then divide by meiosis II, and we will get 4 total products for sperm. Each are haploid. In oogenesis, however, this germline cell that's present in the ovary undergoes meiosis I, and notice that the cell products are not equal in size. The polar body is very close to what we call the secondary oocyte. They're usually attached to one another, and there's very little cytoplasm in the polar body. But we still have 2 cellular products, and each is considered haploid. Now, if a sperm and an ovum undergo fertilization, then both of these cells, polar body and the oocyte, will divide by meiosis II. In the end, you'll get this small polar body producing 2 little polar bodies, and this oocyte, or ovum, whichever you wish to call it, will divide unequally to produce a small polar body and the final oocyte. Eventually, the polar bodies are shed. So is the zona pellucida, which is kind of shown here surrounding the oocyte. And you get a mature egg that is considered haploid. There are some animations from McGraw-Hill available to you that I will also post on YouTube and on Blackboard to help summarize this process.